Hello guys, as I already told you that now I will change my teaching pattern. So I am teaching in notes format. Okay, so let's start our short notes part 3. So let's start. If there is changes in strains of virus of same species, it happens due to antibiotic resistance. Okay, and remember two more points from this. It especially occurs in patients, like when the virus is active inside the patient. Okay, and can be seen in case of staphylococcus. Okay, now our next point. I hope this point is clear. See, if there is any change in the strain of virus or sometimes bacteria also, okay, of same species in that host body, okay, it usually happens because of antibiotic resistance. Okay, now our next point. Secondary infection occurs after a different previous infection. Like a person is already having some type of infection and some another type of infection happens, okay, or occurs. So that is secondary infection. Now let's come to our third point, that is related to our anthrax bacteria. Okay, for this, first of all, okay, it lives in soil. It lives in soil. Okay, it is usually caused by, as you can see, it is usually caused by, like if you are working in pig farm, okay, pig farm workers, or like wool collecting, okay, or like in wool industry, okay, wool collecting, like you are working in leather industry, okay, you are working with cattle or in cattle area, okay, it is caused by at these places usually. Okay, so it is gram positive capsule forming spore forming non-motile, and you know that capsule, it is made up of that polypeptide D glutamate. Okay, now as you know, its vaccine is STI vaccine, and this STI is our live vaccine. Okay, live vaccine. And the test we usually perform for this anthrax is our thermoprecipitation test. Okay, this thermoprecipitation test is also known as AKA means also known as Ascoli's test. AK is mnemonics that I usually use, like, okay, like short form. So AKA means also known as, you know, this in this thermoprecipitation test, it is also known as I told you, Ascoli's test. And in this thermoprecipitation test, there is ring precipitation is used for this, okay, for this test. All right, now this anthrax bacteria, this causes ulcers. As we know, it causes ulcers, and these ulcers are also known as, AK means also known as, black ashar on skin. Okay, now one more point. See, if we took the touch smear is a type of smear, okay, that is touch smear. Okay, if touch smear from animal organ is taken, it appears as streptobacteria. Okay, if the touch smear from animal organ is taken, it appears as streptobacteria. You know, touch smear is a type of smear, this touch smear. Okay, and As I told you, it is spore is stained by a juice key stain. Okay, and capsule is stained by over Berigin stain, all right. And remember, anthrax in touch smear appear as streptobacteria. Okay, anthrax in touch smear appear as streptobacteria. Now next part, as we know, like in our body, if some antigen is enters, okay, so first of all, like there is antigen presenting cells, like they will present that antigen, okay, to our monocyte. Then those monocytes form macrophage, or they will call the macrophages. Okay, and this macrophage is first step during infection. Okay, this whole thing, basically this macrophage forms, this is the first step during infection. Okay, and presentation of antigen to T helper. Basically what happens, antigen presenting cells show them to monocytes, then monocytes form macrophages, and those macrophages then take them or show them to T helper cells. Okay, now as we know, like in our previous video we discussed, like IgM is for acute and IgG is for chronic. Okay, like if in blood you will find IgM, that means there is acute infection of something. Okay, whatever, the infection is acute, and if you found IgG, that means chronic. Okay, but sometimes what they will give you in question to confuse you, they will give them together, okay, and ask you which will decrease first in our body. So remember, if IgM and IgG are written together, okay, so IgM and IgG antibodies decreases firstly in our body, okay, this you should write, like if they ask which antibody decrease first. Okay, now our next part, if in patient you will found like any four known specific prevention of flu epidemics. Okay, so if you don't know like which type of epidemic is this of flu, okay, so in that case we will give rimatidine is given. This rimatidine is antiviral drug for treatment and prevention. Remember, this is an antiviral drug, and it is for treatment and prevention. Now our next part, here we will discuss about like some tests, okay, that are commonly asked. So first one is our agglutination test. Okay, in this test usually what happens, the antibodies will clump. Okay, imagine they will clump. See if this is antigen. Okay, so antibody does what? Antibodies will go, and suppose these are antibodies, this Y. Okay, suppose these are antibodies, this Y. Okay, so antibodies will go and form a clump of them. Okay, like this. Like these are antibodies, this Y, and they will form a clump. Okay, so in this what happens, there is clumping of antigens. So let's write about agglutination test. Antigens clumping under specific, obviously those antibodies which are specific for that antigen. Okay, so under specific antibodies, AB or antibodies. Okay, antibodies. Agglutination test is like type of serological test. Okay, in this electrolytes is used, and it helps in identify antigen structure. It helps in identifying antigen structure. Okay, now next one, heme adsorption test. Okay, in this what happens, see the antigen, as we know the antigen, some antigens, okay, they are adsorbed on RBC. as it is very clearly indicated in the name. So what happens, see, if suppose there is RBC, so some antigens absorbed on the RBC, and those antigens are usually like influenza or para-influenza, okay, influenza and para-influenza, so they are absorbed on the RBC surface. So we took those RBCs, okay, and we put them in like 0.5% solution of guana pig, and we do the test, okay. So this is only important for you, heme adsorption, in heme adsorption test, that antigen is absorbed on RBC, and further we will do test on that, okay. Now the third one is our precipitation test. Precipitation test, okay. In this, first of all, I want to tell you, in this also there is like electrolyte is used, and it usually is used to identify the blood is of human. Like suppose someone gives you blood and asks you like identify that this blood is of animal or not, or like humans or not, okay. So this is used to test like blood is of human or not. So used to find blood is of human or not, okay, or not. In this, what happens, antigen, okay, sedimenting under immune or serum antibodies. AB means antibody, okay. So actually in this what happens, suppose we have some soluble antigen, okay, and suppose we have homologous antibodies. Homologous means they are of antibodies of same type, okay, they are effective only for same antigen, okay. Suppose we have homologous antibodies in this test tube, okay, in this test tube we have homologous antibodies, okay, and we do not know the antigen, like what antigen is, but we know what type of antibodies we have. So we put this soluble antigen in these test tubes, and then in which test tube there will be reaction, okay, there will be reaction like that, X soluble, X means we don't know, okay, so X soluble antigen plus homologous antibody, and in whosoever test tube they will react and form precipitate. Suppose they will react in this B type, okay, and they will form precipitate, okay, and those precipitate then sediment, okay. So in which test tube they react together, so from there we will identify which type of antigen we have. How we can identify which type of antigen? Because we have homologous antibody, we already know which antibody we put in which test tube, so only those antibodies will react to those antigen for which they are made, okay. From that we will know which type of antigen is that. Obviously when they will react together they will form insoluble PPT. PPT means precipitate, okay, insoluble PPT means precipitate. Now next one, indirect immunofluorescence, CNCS, indirect immunofluorescence test, okay. Indirect immunofluorescence test. See, immunofluorescence test is usually done to identify the types of antibodies, okay, in the blood serum, okay. It is used to identify the type of antibodies, and it is of usually two types, like direct or indirect, but we usually use indirect because it is good test, okay. It shows very much specific antibodies about in detail, okay. So we usually do this indirect immunofluorescence test, okay. Remember 1.48, luminescent in this test, okay. So luminescent serum against human, AB means antibody, should be used while fixing smear with investigated serum, okay. In this, while doing the test, okay, in this test the luminescent serum is used against human antibody, okay, while fixing smear with investigated serum, okay. Now in this indirect immunofluorescence test, basically what happens, there is a little bit longer process. Like first we will react those antibodies or to the immune system, okay. Usually this indirect is used to test autoantibodies, okay, autoimmune antibodies. Indirect immunofluorescence test, this test is usually used to test autoantibodies. Auto means, you know, autoimmune antibodies. So in this, first antibody is reacted to the human organ, and then that is taken, and then we do further processes, okay. Now our last part, allergic reactions. Which immunoglobulin you will found in allergic reactions? Ig, E. Okay, and in these allergic reactions, what we will do? We will give antihistamines. Okay, we will give antihistamines. So antihistamines are given. IgE antihistamines are given. So that's all in this video. We will continue our notes in further next video. Thank you.